So I guess with that, I would start to introduce our speakers for today. So I think we go in the order of Allyson. First, I think Allyson would be speaking. So yeah, so Allyson has had an interesting educational background where first she discovered ancient Mediterranean civilization during her bachelor's in Texas, and after which she moved to the UK for her master's degree in computational biology and at the University of New York, and then subsequently her PhD in computer science at the Newcastle University. And since 2015, she is at the Oxford E-Research Center, where she's working on the FAIRsharing project now. And we are very excited to hear a lot about this interesting resource. But another point that I would want to add is that she is very involved in the outreach and very active as a STEM member. So thank you, Allyson. And please share your screen, and we're looking forward to your talk.
Thank you. Please let me know if the screen is okay. Thumbs up.
Yeah, it's all good. Thank you.
Lovely. Thank you for that lovely introduction, Aastha. That was beautiful. I really appreciate it. Yes, I enjoyed history, but I figured it would be harder to find a job in history, so I moved into the sciences, which I also loved. But that was fantastic, a little insight into my history. I do use that actually a lot when I'm working as a STEM ambassador and talking to school kids about how you can have very varied backgrounds and come into science. So today, though, I won't be talking about ancient Mediterranean civilizations. I will be talking about FAIRsharing, which is a resource, a registry of data standards, databases, and data policies. And we're really out here to help advertise your resources and to help researchers find them. So today, I want to just cover very briefly what FAIRsharing is and how we can help you. And that's going to be the whole scope of today, with hopefully some time for questions at the end. So we're a global resource. We're also across all research areas. We are agnostic about the type of research data that you have or the type of research data standard you're describing. But what we want to do is really store metadata about your resource. So we store information about data policies, community standards, and repositories, as I said, and not just the information about the resource itself, but also the connections, the relationships, the ecosystem, you might say, that surrounds every bit of a digital object in the sciences. And we have two main ways that people enter and interact with FAIRsharing. On the one hand, we have consumers of FAIRsharing, and I'm going to tell you a little bit about those here. Consumers of FAIRsharing who wish to search our website and find a resource that they want to use to store their data or to structure their data. We also have producers who come to FAIRsharing. They're people who develop the databases and the data standards, and they wish to describe those resources with us. We have also a lot of different stakeholder groups that I'll go into a little bit more detail in just a moment. But I wanted to show you that in general, the vast majority of our records in FAIRsharing describe databases or data standards, though we do have a variety of other ways that we can slice the data for specific communities. Very briefly, our different members of our community encompass a lot of different roles within research. We have researchers themselves who are looking to identify a resource to use, maybe incorporate it into their data management plan. We have research data facilitators, such as data stewards and librarians, who are using FAIRsharing not only to help those researchers, but also as a foundation for training material. We have funders that recommend the use of resources through us. We also have developers of resources, as I said. Groups like yourselves come to FAIRsharing to create a particular slice of FAIRsharing just for your community. And you guys have already done that, and I'm going to show you a little glimpse of those particular records later on. And then finally, journal publishers, in the same way as funders, come to us to try to show the authors that might come to submit manuscripts with them what resources they recommend. So what does a fair sharing record look like? Some of you may already be very familiar, and indeed, I'm sure some of you are familiar with fair sharing, but for those who aren't, a short overview of what you get when you come to fair sharing is next. So first of all, we have lots of different ways you can describe your data standard, your policy, or your database, but you don't have to use them all. You use what's appropriate for your community. And here's just some examples of the metadata fields that you can use in fair sharing. And the example I've given you here is the image data resource, which is part of the Euro-BioImaging collection in fair sharing. And records are citable, okay? So it's not just the record itself in fair sharing or the resource publication, the publication for the image data resource in particular, but we have different ways of attributing you guys as the community experts. So right now, the image data resource has 4 different community maintainers who have accounts with us and who log in and make sure the record is up to date. And we allow them to link their ORCID to their account so that people can find them really easily once they leave the fair sharing site. We also provide a very high profile view of which publication IDR recommend. Oh yeah, sorry. And we also provide a DOI itself for the record in fair sharing so that you can cite your description of your database with us in fair sharing using a DOI. We also have relationships. I mentioned those a bit earlier. We have this network that connects all of the records within fair sharing. And this is probably my favorite part of fair sharing. And so what it does is first, we start by just listing here the related standards, such as gene ontology, the related databases, and also any data policies that recommend the use of your resource. So these are a snapshot of the policies that IDR is recommended in. We also list those collections, those slices of community views over fair sharing that your resource is in. In this case, IDR isn't just a member of the Euro-BioImaging collection, but also of the EOSC Life and the OHM collection. We also monitor the evolution of a resource. So if you take this snapshot of the Euro-BioImaging collection, you can see that 3 of the resources here have green circles. That means they're ready. They're ready to be used. And one of them we've marked as deprecated. And when you look at that record, we don't just delete it if it's no longer available. We mark it as deprecated and we give a reason. So for instance, when we migrated all of our data across to the new version of fair sharing that's just appeared on your screens this month, we checked all of the database resources, and those who had broken links and who didn't reply to our queries got marked as deprecated. And indeed, this is one of them. And it's not just the life cycle itself, which we have a number of different tags, as you can see here. It's the fact that our curation team checks every single record in fair sharing. We aim to do it once every year. Sometimes it's a little bit longer than that, especially with the recent work we've done to create the new website. And anytime someone edits a record, it gets moved to the curation team to double check to make sure it's within our curation guidelines. So I talked a little about those relationships, this ecosystem of records within fair sharing, and I just want to show you why those are important. I've shown where they are visible inside a record, but that hasn't really shown you their connections. So I'm going to take the Euro-BioImaging collection, and I'm going to show you a little bit of how these relationships work using that collection. So here again is the view of a portion of the collection that we have for Euro-BioImaging. And this is what it looks like when we look at the graph view. If I just go up one, you can see on the left-hand side of the screen, there's a small button underneath the general information that just says View Relationship Graph. That's how we get to it. And so here's the relationship, the relationships from, let me start again, the members of the Euro-BioImaging collection. So those resources that the collection groups, and also the relationships among those resources. And every time you click on a node in the graph, you'll get its particular neighborhood displayed. So what does this show you? Well, it shows you how Euro-BioImaging members are related to each other. You can see there's a small node on the bottom right around IDR that shows some of those relationships, but that in general, this collection has members that don't have a whole lot of relationships listed yet. So how can it be improved? Well, we can add all the connections to your database that your database might have to other data standards, databases, and data policies. And we can add connections across those member resources within the Euro-BioImaging collection. I give you a little bit of an inset view of the IDR here and show that when you hover over individual parts in the relationship graph, you can see the relationship specifically for those nodes. Have a look at another graph. This is Dryad and is clearly a denser graph. I thought I'd show you that in comparison with yours to show you what another graph looks like. So this shows you the standards Dryad implements, and those are shown with the circles. It shows the repositories Dryad has the relationships with the squares, and it also shows the policies, which are there as triangles. And the different types of relationships are shown in different colors. So with my last few minutes, I'm going to just do a very quick run through of how you create and update a record, and then a final summary of how it can be used. This is going to be a very short overview, so I welcome questions later or emails in the end. So you can create an account with us or you can log in. There's a various number of authentication methods that you can use. Now, before you start creating a record with us, we appreciate it if you search and check and see if it's already there. So here, someone's searching for the Data and Service Center for the Humanities. They find it, but if you don't find it, there's a button at the top of every page called Add Content, and that will get you started on the creation of a new record. So here we have Dash. We found the record. What do we do next? Well, we create an account, as you saw before, and then we choose from the action menu, Request Ownership. And then that sends a message to the curation team at FAIRsharing, and we double check that you're the right person to update the record. And for that, at the moment, if you wish to be a maintainer, you need to be a developer of that resource. Once you're accepted as a maintainer, you have access to the edit interface, and you can move along the series of tabs at the top of the page, updating the metadata and adding anything that might be missing. One particular feature is that we have a number of controlled vocabularies and ontologies. Indeed, our domain ontology crosses 50 different external ontologies based on what our FAIRsharing users have needed and asked for in the past. And so you can search across all of these vocabularies, adding the right tags and terminologies for your needs. You can also add these relationships like I've been telling you about so far. So the way you do that is a separate tab within the edit interface, and it looks like this, and you can choose which one you want by searching on the left-hand side and then adding it to your record on the right. And you get an option to choose those relationships that might be relevant and how indeed that resource is directly related to you. So for Dash, they implement the ARC identifier schema. And so we add the type of relationship as implements for that, and then it appears in the record as part of that relationship graph. So how do people use FAIRsharing right now? Well, I talked to you before about collections, and Euro-BioImaging has a collection with us. Here are some of the other example collections that we have. We have one for astronomy with the International Virtual Observatory Alliance. We also have GBIF, who have maintained a collection with us. These are branded pages that allow you as a community extra visibility, not just for the resources that you are a part of, but for the organization you represent as a whole. Sorry, the community you represent as a whole. I want to show you a couple hairballs. I think no network-based slideshow is complete without a hairball or two, but I wanted to show you a couple of different ways these collections are implemented from researchers themselves rather than from particular communities. So I have two examples. The first is a resource collection of over 500 COVID-19 related standards and repositories that have been linked to a particular paper that was made by the RDA to talk about which COVID-19 resources should be used. And so this presents a live view with life cycle. So it tells you if there are any in development standards. It tells you if there are any deprecated standards since the paper was first come out, and it gives you a nice live snapshot. Here's a similar hairball, but it's for an ISO standard. So rather than particular authors from a manuscript, this is a particular authors of a draft ISO standard. And so having this live collection on FAIRsharing allows everyone who comes to the ISO standard not only to see a version that is something that you don't have to pay for because the ISO standards have a cost, but also that allows you to get a live version and really work with and manipulate the relationships among the records. We also provide an API, and there are a number of FAIR enabling services that access FAIRsharing as part of their larger tool. And so they use this to help inform tools that are relating to data management plans, data management guidelines, FAIR assessment. And also we're part of a broader data discovery service that we're working on now to really help third parties find out how programmatic access can happen to various databases and data standards even through their record in FAIRsharing. So we have collaborations with OpenAIRE and a number of other resources to access that discovery, those access points. A brief note about policies. I told you that funders and journals use us to create slices of the data. This is an example from PLOS, and this is the PLOS recommended repositories. And publishers use FAIRsharing to list those resources they recommend and also to help inform the creation of those recommendations. FAIRsharing itself, well, we also implement FAIR in our own small way. We have a number of schemas and standards that we use to encourage findability both programmatically through things like schema.org markup and JSON export, and through the web services. We also have an open license to allow you to use the data and in any way that you see fit, including the code. A majority of the code for FAIRsharing itself is also available. We're also integrating with OpenAIRE and with ongoing work with integration with the ROR and FundRef organization databases. Finally, I just want to say, how can we help you? This is sort of the key. Out of all this I've told you, we really love having your input. We love having your resources registered with us to help you be more fair, to help your users, the people who come to you, find you more easily and help them make their data more fair. We maintain the records as best we can as a curation team. We're jacks of all trades though, and you guys are the experts. Yeah, so you know more about the key features and the descriptors. You know more about the landscape and the connections between these resources. You guys are the experts, and we want to attribute you, and we have some interesting plans on the work to how we can attribute our maintainers even more than we do now. And we'd love to hear about any way that you might be interested in using FAIRsharing or contributing, and indeed any way that you might wish to be attributed. And I finish with the required slide of all the fantastic people who have helped us develop and use FAIRsharing and lead it in the direction that it's been coming to for a long time now with its brand new website and all of the really exciting plans we have for the next 6 months now that the new website is out. I want to finish just ultimately by saying thank you very much. The homepage you see listed there is our stable URL. You can find the new website at beta.fairsharing.org, like I showed in my very first slide. And over the next few weeks, as we work out the final kinks in the beta site, we will move everything from the beta site to this stable URL. That's everything I have for now. If I stop sharing, and I've deliberately moved reasonably swiftly through those slides so that if anyone has any particular questions, for instance, about the Euro-BioImaging collection or about making or searching records on the new site, then I'd be happy to do that. Aastha.
Thank you so much, Allyson, for that fantastic talk. And of course, we all work very hard towards open and fair science and open and fair resources, and FAIRsharing is really important for us to get that visibility. So I think it's very important, and thank you for sharing it with us. Now let's look at if we have any questions. So I would say it's Arina first. Arina, please go ahead and ask your question.
Yeah, thanks a lot, Aastha. Allyson, thanks so much for this wonderful overview. I learned a lot about FAIRsharing now, and I find it really very interesting how you try to see the connections also between the areas and to link with people. The first thing that came into my mind is how well connected are actually astronomy fields with image data resources, because in principle first of all, it sounds okay. It's two different areas, but the approaches that are actually used in astronomy for image analysis, data analysis, are actually quite similar to imaging technologies on microscope, right? So I was wondering how well these two fields are interacting.
That's a really great question. I love it. I wish I could say we had a lot of interconnections between astronomy and imaging, but I'm pretty sure the astronomy standards, from what I've looked at myself, are pretty much in an island of their own, an island of interconnectedness. So if you know of any imaging standards that are used by both fields, I would really, really love to have that in FAIRsharing and have those links made obvious and made clear, because I think you're right, there is a lot of overlap, and I don't think we yet include that. I know the person who has developed the IVOA collection, the astronomy collection, and he did a lot of work. He came to us and said, I'd really like to do this. And he maybe made 12 different records. A lot of those he built up to create the collection almost from scratch. And he did a lot of great work, but I don't remember him including any imaging standards specifically. So yes, please come and add any that you've got.
Yeah. Personally, I'm not from astronomy fields, but we have an institute close by, and there's astronomers here. Maybe
you can ask them.
Maybe we should somehow connect with them a bit more and spread this information on FAIRsharing too.
Absolutely.
Great. Thank you very much.
Thank you for that interesting question, Arena. Indeed, we should build more bridges. Okay, next question is from Antje. Please go ahead.
Yeah, thanks a lot, Aastha, and thanks a lot, Allyson, for this wonderful presentation about FAIRsharing. So Euro-BioImaging is involved in BioCovid, and actually we are having a task coming our way regarding the mobilization of non-human data. And in the first meetings, FAIRsharing was mentioned as the goal where we should bring the mobilized data. So what would be your expectation or task that would you give to us at the moment, where you want us to go in the next 2 to 4 years?
Oh, well, that's really good. I do know through my boss and through the emails I've seen, of course, FAIRsharing has its own part to play in BioCovid. And so it sounds like we're all members of the same grant family, which is really nice. So I think that what we need to do is just find out how we can represent the resources that you use and that your community creates, and see if there's any gaps in what we've got in FAIRsharing. And in terms of BioCovid, I think we just need to have a look at that particular part of the grant, because I'm not that familiar with our particular. I know that a lot of our BioCovid work is going to be integrating more closely with OpenAIRE. And so we've been talking with them about getting all of our objects visible through the OpenAIRE route. So I don't know how much Euro-BioImaging uses OpenAIRE, or indeed if you have any various, they mainly are available for programmatic access and searching. So it provides a new route for people to find FAIRsharing objects. For instance, OpenAIRE doesn't store any information about data standards yet, but as soon as the integration with FAIRsharing is complete, they'll suddenly have a whole new suite of objects available to their network graph that they didn't have before. So I don't know what the right answer is to say, what should we do for the next 2 to 4 years between BioCovid for Euro-BioImaging and FAIRsharing? I wish I did, but I think that sounds like the great start of another meeting.
Wonderful. I have a second question, if okay, with everyone here. So it's kind of quick. So FAIRsharing is a community resource, as you say, and as it's also written on the website. But is there any institution behind that has been funding or is funding FAIRsharing?
We've had some funding from various places. We are a University of Oxford project, so all of us are members of engineering science department. I believe Wellcome Trust has provided, we've just got some new money from BioCovid. In the past, we've had some funds from as diverse locations as Japan and the US. And we've also had some recent funding from, I can't remember the list. This is a good question for Susanna. There is a page on the website, I think it's on /communities, and it has a list of all of our funders. So I feel like I'm failing my quiz. Susanna would be doing this to me right now, but I've given you a short summary, and there's quite a lot of European funding as well. Not just things like Wellcome from the UK. So I have to say there's definitely European funding, and I can't recall which ones. So
thanks a lot, Allyson.
thank you. Have a look on the community pages anyway, and it'll have everything. And please do ask if you have any questions.
All right. So as we are waiting potentially for more questions to come up, if somebody is still thinking and formulating theirs, I can go ahead with mine. I was actually wondering that since it's a curated database, does a collection or a policy need to qualify in a certain way to be accepted, to be registered, to be mentioned on the FAIRsharing resource?
Yes, so collections, we have a variety of collections. Some were developed in the early days and they're more generic. I think Egon Willekagen made a chemistry collection. Sorry, Mariana? Oh, you? Oh no, okay, it's fine. If you were just saying something, that's fine. It's no problem. So I know he made a chemistry collection, and that was just generally designed to help people find good quality chemistry resources. But we're moving away from that sort of generic collection because in the next few weeks, we're going to be showing a new route into FAIRsharing, which is browsing the research area hierarchy. So we have these subject tags. And so one of them will be chemistry, and one of them is imaging, and things like this. And so we're going to have people able to browse through the hierarchy and display their own live collection based on the subjects. So we don't really need those particular collections anymore. Collections usually come to us from community organizations like yourselves, or authors of manuscripts, or funders and publishers. So they have a particular reason to showcase a particular subset of resources. And we're very open to any kind of collection, but we don't usually, for instance, offer them at the individual researcher level. Like my collection of interest wouldn't be a collection that we would keep on FAIRsharing, though one of our future projects is to develop a route so that users can create and store their own slices, their own mini collections that may be only visible to them. So it's a good question. And policies, again, as long as there is a document that is approved and by the community for which it is a part, like a funding document or a author's instructions document or a community document, then they have a policy for how data should be used, then we're happy to have a data policy document. A number of our data policy records don't have any relationships to any of the other records in our registry, because they generally say things like, we wish you to use any database that has an open access license. And of course, we can't easily, we can point people in the direction of those resources that have the right license, but it might be in completely the wrong domain, or it might be not quite the right kind of open access license. And so some people's policies are more vague than others. So we endeavor to include those records, but they won't have any links to other resources. So it's basically, as long as you're sensible, we want you in. It's a pretty open policy. We like having some basic metadata and some basic information about contacting and licensing. But if you have anything that's of use to a number of people, then we're happy to include it.
Great, thank you so much, Allyson. I do not see any other hands that are raised, and I think the chat's empty for now as well. But of course, the discussion can be taken up in the chat. So please go ahead and keep putting in your questions. And Allyson, if you have time, just keep an eye on the chat, and that would be great.
I will. Thank you.
Thank you so much. All right, that was very exciting. Now let's move on to the next talk. So the next talk is from Beatriz Serrano-Solano, and she is a community and project manager at the European Galaxy team at the University of Freiburg. She studied computer engineering and also did her PhD in health engineering from the University of Malaga. Then she was working at EMBL in Heidelberg as a postdoctoral fellow, and she's worked previously with Euro-BioImaging, as well as she was working on the project of a European Open Science Cloud, so the EOS Live project. We are going to learn some new things that she's been working on related to Galaxy and image analysis, bringing image analysis flavor to Galaxy. So Beatriz, we're looking forward to your talk, and please take it away.
Yeah, thanks, Aastha. I'll try to share my screen now. Okay. Should we go now?
Yeah, looks good.
Okay. Great. Thanks, Aastha, for the introduction, also all of you for joining today this virtual pub. I would like to give a brief overview about the options to perform image analysis in Galaxy, including also some practical examples developed at EMBO when I was there within the EOS Live project, as Aastha already mentioned. So I guess the first question for today is, what is Galaxy? So Galaxy is a platform for performing computational analysis that serves as an abstraction layer to an HPC or a cloud environment. So users really don't need to deal with command line at all. They have a graphical user interface and all the operations that you can perform in that interface, you can also do with an API, so programmatically. There are many different components that can be plugged into the platform. So on one side, you can bring data from different sources. There are tools that can be integrated there. They are changed and create workflows, and you can also visualize data. The analysis can be automatized with these workflows, and Galaxy can be plugged also into different RDM solutions. So on one side, on the upstream level, can bring data from different DIM systems, or afterwards can be also to deposit the outcomes of your analysis in a repository, for instance. It's an open source project, so that means that everyone can contribute to it, and all the code is available. It's also a global project. There's also open infrastructure, and there are at least 3 main continental servers, one in the US, another one in Europe, another one in Australia. We are aware of 165 public instances so far, but it's sometimes very hard to track, so we don't know where that is, because sometimes it's in industry, sometimes it's in research institutes, so it's hard to get those numbers. And there's one imaging instance also at EMBO that it's managed by E and Chariman. And it's also a global community. It's a big community of users, of tool developers, admins, and trainers everywhere. Okay, so this is how Galaxy looks like. You are seeing now the interface in Galaxy Europe. So on the left panel, you see the different tools that you can select to perform your analysis. They're classified by topic. Then there is a central area that is the working area where you can input the information, the parameters that you want to tune, or you can also visualize the data there. And then there's the right side, it's a history. So history is a component in which all the operations that you are performing are appearing in order of execution. And that creates a workflow, but it's kind of a way to document all your work, like you would do in wet lab with a lab notebook. Okay, so this is a short video how it looks like live. We have had this imaging flavor in Galaxy Europe, and there are several resources there. You can see different training materials, so introduction to Galaxy, and for all the imaging community who is interested in segmentation, feature extraction. There's also some on mass spectrometry, and you will find there slides, hands-on materials, small data sets to work with, and also workflows to perform the analysis. There are different tool suites there for data accessing, for segmentation, feature extraction, different biological areas, and there are more that are being integrated right now. I will talk about this later on. So this that you've seen here, it's really not the main interface of Galaxy Europe. So it's a customized flavor just for image analysis. And that means the tool panel that I show on the left side before, it's customized just to perform this kind of operations. Okay, so let's start by the first step of the image analysis, is getting the data into Galaxy. And there are different ways. So the screenshot that you see here is a upload tool. You can get the data from your computer, but you may have that in some other repositories. Like for instance, you can have it in an S3 bucket, or you may need FTP to transfer the data from somewhere else. There are different protocols that are supported there, and it can be easily extendable to adopt to new ones. The other way that we were using within EOS Live to get the data, in this particular case from the IDR that was already mentioned by Allyson and FAIRsharing. So the IDR contain different subsets of images, and we created within the Corbett project in collaboration with Euro-BioImaging this tool. And the tool, this is what you would see in the central area, so in the working area of Galaxy, where you input all the parameters for this tool. And you see that you can pick their IDR if this is the source where you want to get the data from, but you can also get data from a local Omero instance. You just need to input your credentials in your user preferences, and that's it. Then you can pick which images you want to download. So you will need to get the image IDs from the IDR and upload there in the form of a file or just a separate list, whatever format you input that there, and those are the images that you bring. The other part would be to define the region of interest. So sometimes you don't want to bring the whole image, just want to crop it before, take a particular channel, set playing, or somehow customize what you want to bring and not bring the whole thing. And that's also possible with this tool. In terms of imaging tools, so I've mentioned already the IDR Omero download. There are different tools for different disciplines, and to include a tool in Galaxy, all you need to do is to create an XML file defining some metadata, like the inputs, outputs, which command will be running on the back. And with that, you can have it already available. This is particularly useful for the imaging community in terms of collaboration with other fields. So you can have now analysis there that are combining other disciplines that are already using Galaxy quite a lot. There are some tools that are coming soon or are almost there already. We were working mostly within EOS Live in Cell Profiler. There were some image tools already there. But soon we will have a new version of Cell Profiler that's, I think, is very close to the end. Cell Pose, it's also there. There is container, it's available. There are other different tools that are for different type of operations, 2D, 3D, that will be also included soon. Okay, and this is something a bit different from what you've seen before. So now we are talking about interactive environments that you can connect to your automated part of the workflow. And in this case, I'm highlighting here a few that I think can be interesting for image analysis. It is Jupyter Notebooks and RStudio. You can also have as part of your workflow, and also virtual desktop environments. So all of this gives you the flexibility to perform a particular analysis in case the tools that you needed weren't there, or to maybe filter at the beginning of your workflow some of the information that you want to process, or to analyze the features at the end. And now I will show you an example of a workflow that we were using in EOS Live. So we wanted to analyze the nucleoli in different RNA screens or expression or compound screens that were available in the IDR. So we selected a number of screens, in particular the DNA channel that you see in the image here in the right side. And we wanted to identify where is the nucleolus there, which position, if there are, how many there are inside each one of the nucleus. So you see that the area without DNA, so the absence of staining, is actually the nucleoli. We wanted to know how many we find whenever a gene is silenced or expressed or a compound is acting on it. Analyze also the shape, if they were having a different distribution within the nucleus. So we wanted to have that, and we selected a number of data. In total, I think it was like 200,000 images that we wanted to analyze. And we perform a workflow like this there. So first we assess the data, only taking the region of interest, or of interest. In this case, it was the center of the image, and only some images that were already annotated in the Human Protein Atlas. We use the IDR download to bring these images into a Galaxy history. And then we use Cell Profiler, and that was part also of the project in which my colleague was working, integrating these different models of Cell Profiler in Galaxy. And with that, we performed segmentation and feature extraction. And then the last part was a bit more flexible and wanted to have an interactive environment for analyzing the features. And this is how a workflow really looks like. So each one of these boxes that you see in blue is one of the tools, and they are all connected. So the data, it's going through all of them. This is an example that is deposited in the workflow hub. So we have this workflow that I showed before in the workflow hub, but also afterwards, after the project was over, Chariman and Isung were working also in integrating some tools for object tracking, and easily, we needed to adapt a few modules, but we could reuse many of the previous ones and create this object tracking workflow as well. I will show maybe results later. So this is now a feature that it's integrated into the workflow hub. So you can go there, search for a workflow that you're interested in, and run it on usegalaxy.eu. Just clicking on the button will send you back to the European Galaxy instance. Okay, so this is a feature that not many people know about. This is a workflow report. So once you've performed your analysis, you can, with Markdown, create a report in which you're explaining all the steps. You can include some figures and information that you've been generating all over the way. And you can also export that as a PDF and share it with your colleagues. Okay, in terms of computation, so I mentioned already the public servers, and so this is an open infrastructure, and the point is that anyone can use it. So the point of the Galaxy project is also to provide these environments for reproducible, transparent, and accessible research for everyone. There are also private instances, but the main ones are public, and the interface is user-friendly, so you don't need to code, you don't need to deal with any technical side on the cloud or HPC, you just need to use this interface, but you can also access that programmatically and do the same operations as you do in the user interface. There's now some modernization going on with the API, so now it's been transformed to fulfill the open API standards. This provides a framework that is interoperable, so you can actually create your workflow with a combination of different image analysis tools. So maybe it might be that you like having a particular operation done in ImageJ, but then you're also interested in Cell Profiler for different kind of features. You can combine all of them in just one single workflow, and then have an interactive environment, some machine learning tools, and all of that you can make in a very reproducible and transparent way. You can have your workflow, you can share it with other people, and enhance the reproducibility with that. So the infrastructure in general, it's assembled in a scalable way. There is a Pulsar network in which one can compute some parts of your workflow in different nodes, so in a completely different location. One can also perform the analysis in a place where the data is. So there is a network in which one can submit the jobs to different locations. Okay, so from the perspective of imaging facility, for instance, it's also important to give support to the users. So there is a centralized way of aggregating all this information that users can submit. So whenever a tool fails, there is an option in the interface to submit a bug report. And this is what the person on the other side would receive. Links to all the data sets, to the history, a message from the user if they input something, all the information about the tool that was used, the version, everything, what was the command that was run, all of that. It's provided so that the error can be reproduced and fixed. And yeah, so that provides also scalability, because there can be many users submitting this, and it's all done in a centralized way. But there is also community support. So whenever people are having problems or they don't know how to do some particular analysis, they can go to a forum that is called Galaxy Help. And there, people are asking questions and the community are replying. There are also people that are just giving support to the community in general. There are plenty of page views. You can see 1.3 million and many new users in 2021, many posts as well. This is just this year. Okay, then training, it's also important. So this is the Galaxy Training Network. It's a repository in which all the training materials are deposited. There are different categories here, so I will highlight some that I think are important for the images side. There's a Galaxy for Scientists. You can find all kinds of disciplines there. There's one for imaging, and I will talk about this a bit more in a minute. I wanted to highlight also this new category that started only recently, 1 or 2 weeks ago. This is data science one. Maybe that's also interesting for you. There are some trainings about different technologies there. And there is also even tutorials for knowing how to contribute, or tutorial to how to create a tutorial, basically. Yeah, so the imaging category has now 4 different tutorials. Some of them were related to the work I've shown you before. One is a feature extraction and segmentation for nucleoli, and the other one was the object tracking. And you see how the tutorial will look like in the right side. So you have an introduction with all the biological explanation, and you have step-by-step guided tutorial. This is the tracking tutorial and workflow that I mentioned already. This is a dividing nucleoli Drosophila embryo, and we also develop a tutorial to do this. So you see the input on the top and what is the outcome of this tutorial at the bottom, in which the cells are being tracked while they are dividing. Okay, so also training infrastructure. Okay, so this is also a service that is provided in the public, so far as in the 3 main public instances. So basically, it's a special queue for people that are doing a training on a particular date. So whenever you want to run a training with a given tutorial, you submit a request, and we process that and provide you with a link for a special queue. That means that the students do not have to wait in a regular queue like the rest of users. They will have a special one in which the jobs will have priority to run, so that they don't need to wait. And they also will get, so the trainers will also get a link to a dashboard in which they can see which tools are be running at every moment. So to make sure that everything it's going well and that everyone is progressing at the same pace. And this has been proven very useful also for all the online settings recently. We've been running plenty of those, even more with the pandemic, and now there are more than 9,000 students that have been training using that. Okay, so in terms of outreach and training and all this information, we've been running several series of webinars, in particular for COVID, but there was also one for advanced features for those users that feel more encouraged to do more advanced operations in general. And in terms of training with the pandemic, we had many of those online. This year in February, there was the GTN Smorgasbord that had more than 1,200 registrations. People were super happy with this. There was plenty of video that were created. It was all asynchronous. People could have all the flexibility to watch the videos whenever they like, and there was live assistant in chat. We also ran another one on plant transcriptomics also in spring. Over summer, there was another one on SARS-CoV-2. We are going to run another one soon. Yeah, so there are plenty of training materials, videos with captions for all different roles, so from scientists, developers, and also for admins. Another ways to interact with the community, it's a Galaxy Community Conference that it happened in every July. We had, last time was supposed to happen in Belgium, but we had to do it online. And that's a good way also to present your work and to get to know what's going on. We also have PaperCuts Day. We just had that yesterday, and this is a co-fest to have, to get help and to show, to try to improve minor things in Galaxy that can be fixed in a few hours on one day. So people join there and try to get help from experts to do these things. There is also the Galaxy Training Network co-fest every 3 months, in which Same, they will be contributing to the different training materials and addressing different questions. With all the experience that we had over the pandemic with these trainings online and so on, we wrote a couple of papers that are published recently in May, I think. Yeah, so for the upcoming events, we have 2 websites in which that can be checked. So one is the galaxyproject.org, where all the global events are tracked. There's another one in Europe for all the European audience. You can also follow us on Twitter. And I wanted to mention 2 particular trainings that are coming up soon. One is at the end of November. This is a transcriptomics, that is a transcriptome in Spanish. So that will be an experiment for, so training in English, training in Spanish, human translate in Spanish, and automatic translate in Spanish. And we are going to see what is the best solution or how the trainees find that more useful. And as I said before, we are going to run the SARS-CoV-2 workshop again at the beginning of December. This time will be just 1 day, condensed workshop with all the information. Okay, and with this, I wanted to thank from EMBO to E and Giancarin, because they were the main parts of the iOS Live demonstrator that we were running. And in the IDR, also Jean-Marie was very helpful with all these interactions, and Peter from the University of Freiburg, and in general to the whole Galaxy community, because I've only done a minor part of this project, but there is done by people all over the world. Yeah, I'm happy to take questions now.
Thank you, Beatriz, for that fantastic talk. And it's very interesting what all one can do now in Galaxy, and that's very encouraging as well.